Hello, we're glad you've joined us for this live webinar, Patient-Derived Micro Tumor Systems, Future Trends Towards Improved Testing Systems. I am Judy O'Rourke of Labroots, and I'll be moderating this session. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Their innovative detection, imaging, informatics, and service capabilities combined with deep market knowledge and expertise help customers gain greater insights into their science. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Giridharan Periyasamy, PhD, Platform Leader at the Center for High Throughput Phenomics at the Genome Institute of Singapore. Dr. Periyasamy's research focus is on the cancer biology, drug resistance, disease modeling, and signaling pathway networks of human diseases. He is able to run high-throughput functional and chemical genomics for target identification and validation and proof-of-principle studies. Dr. Periyasamy's current focus is on discovery and identification of novel gene functions and phenotypes upon RNAi-mediated gene silencing, or cDNA overexpression, for uncovering novel mechanisms of drug resistance. He plans to develop robust and comprehensive panels of 3D cell culture models, patient-derived primary cells for the characterization of different disease phenotypes, and investigative chemo response to novel or known drugs. He would also like to establish an array of next-generation preclinical 3D tissue models for predictive toxicology screening and validation against traditional animal model systems. Previously, he worked as a scientific director of the HTS-HCS Screening Core Facility at CCAMP, NCBS, India, and as a group leader at Piramal Life Sciences Limited in Mumbai, India. Dr. Periyasamy studied at the Center for Biotechnology at Anna University, where he obtained his PhD in biotechnology. He has more than 15 significant publications and 8 patents to his credit. Dr. Periyasamy will now begin his presentation.
Hi. I'm welcoming everyone to this webinar, which is sponsored by PerkinElmer. I'm going to discuss about the patient-derived microtumor systems, which is a future trend towards improving the testing system for us to realize the potential of HTS and HCS actually, which is high throughput and high content screening. So in this slide, I'm going to discuss about how this evolution of HTS assist strategies as for the past decade, because initially the HTS was largely focused on biochemical based, which is like an enzyme substrate reaction, and people will look out for the readout. I think as the technology improved towards the high content screening, which is like a high content imaging, so we can look at the specific changes in the nucleus or in the tubulin or in the specific proteins. So like when those technologies have started improving, I think this high throughput is coupled with a high content screening with the technology involvement. Actually, you can see that like how deep like people can phenotype now actually, because this is what leads to there is a revolution in the high throughput screening. I think people have started looking back into the high throughput screening and for the meaningful target identification or drug development. So what is the different types of high content screening systems can offer a different type of assays to us? I think here you are seeing equipment which is a Phoenix, which is one of the latest version from the PerkinElmer. We have this in our platform here in Singapore, I think, which we are using this particular core technology by different users of different disease biology groups to run their high throughput and high content screening. So here we can do fixed cell assays, and we can do live cell, and also we can do a complex cellular models like 3D models, and then protein-protein interaction studies. And we can also try, like, basically this will lead to the high throughput phenotyping, because in a well level data, or we can select the cell level data, which can be followed by the deep analysis of Columbus and other related softwares. So there is a need for us to generate relevant in vitro models in order to go back to the translational research, or whatever we do in the bench, if we want a complete translation impact of what we are trying to do. So I think if you see this, the different type of cell culture or the animal model, say for example, if it is a 2D cell culture, or if it is a 3D, or if it is an organoid, you can see the ease of manipulation is easy, because if you wanted to run assay very quickly, and if you wanted to have a very reproducible data in terms of, like, say your ratio is close to around 90 to 95% of reproducibility, then I think that's how the screening was there previously, actually, because a lot of people were in two-dimensional cell cultures. But I think if you see the whatever the output we are going to see in this particular screening effort, actually, because if you see the green line, the more power which we are getting it is only from the highly complex models, actually. I think that's why I think some of our groups here in Singapore, actually, right now we are working on a lot of 3D models as well as on the organoids model in order to derive the more relevant and also the meaningful information which can be translated back into the clinic. So I think to do this, I think we have a high content biology. Basically, like, I can talk about this. Basically, like, you can look into the cell cycle or the tumor cell morphology, or you can look into the skeletal muscle morphology, or like, say, hepatic morphology, or like you can look into the genotoxicity by looking specifically into the nucleus. So based on this technology, the way it has come up right now, so people are now revisiting the phenotype-based drug discovery, because earlier the target-based drug discovery, right from the enzymes, isolated enzymes, and then you look for an enzyme substrate reaction, and then you find agonist or antagonist. Then you come into the cell-based assays. But now, I think now, because since we have this high content approaches, so people are now trying to embrace the biological complexity of creating a much more relevant assays for the particular disease model. I think that is going to be an advantage for every disease model, because we are going to look like we are going to gain the basic informations, which is going to be really useful for the translational effort. I think coming into the cancer, right, I think everyone knows this. I think human cancers are completely heterogeneous. So it is like every thumbprint, how it is different, so every tumor is also different. So people are looking the cancer at a different angle, because it is not like a one disease. It is driven by multiple dysregulated pathways that can lead to the cancer. And when you look into this, the complex disease network in cancer, and also like how we can handle this efficiently, because you can see how these are all interlinked. You can look at this in the two different level, like a patient level, and also at the molecular level. So at the patient level also, every patient is different, because you may not see a response for a same drug for a similar phenotype when you find it and give it to them also, because sometimes the patient response is completely unexpected actually. So this is because of how these some of the major dysregulated pathway or some of the minor pathways which are responding to the drug, and how it is giving back sometimes the relapse, because one of the major things which we are talking in cancer is a relapse and the resistance. So how this cancer genomics is going to help for this, from the discovery of science to the personalized medicine. So basically, when patient comes to the clinic, they'll get the patient consents, and then they will look for their cancer genomics, like specific mutations. Say if it is a breast cancer, they will look for what kind of phenotype it is. So if it is a lung cancer, they will look for EGFR positive or negative, and what kind of mutations they have. So and then they will validate, and then they will use their knowledge to identify the drug targets and the biomarkers. Then they will try to select the drug that might work for that particular patient. So before going into the personalized oncology approach, which we are doing at our GIS in Singapore, I would like to show this the way we do screens, the different type of screens, which will give much more relevant data, which are completely translatable to the animal model. And also, like, we have done one co-clinical setting in which we have seen that the patient is kind of benefited from the guide, from the kind of the decision which doctor has derived from our research work. So I think if we are looking at this image-based screens to determine the therapeutic sensitivity. So here we have two different cell types. I think these are all primary primary cancer-derived cell lines. So they are labeled with a green color dye, and another one is with a TG tomato, which is red color. So say for example, if it is a primary and a met cell lines, we can label them separately. And then what we used to do is we can seed them together in a 384 well plate, and we will do a just image-based screen, and where we can look into the compound which are killing both like primary and met, which may be completely toxic or maybe non-specific. But we have also found some compounds that are specific to red, which is a metastatic cell line. And in the same screen, we found some compounds that can kill specifically primary cells. So I think the basic idea is here to because in the one single screen, we will be able to find out the compounds which can kill specifically to this. And I think we have shown the IC50 graphs here, which can tell you the IC50 difference between some of the selected compounds, and also the bottom two, which shows that this compound is showing this compound is showing an equal activity on human primary as well as human metastatic cell line setup. So I think what we do is actually when we do screens in 2D, and parallelly we have also started implicating we wanted to develop a robust in vitro 3D architectural screens in parallel to the 2D screens, because we have seen whatever the tumors are, which is it will be good, whatever the response which we are seeing in 2D screens, if we translate them back into the 3D model. So we have seen the number of failures in the further validation is completely lesser. So I think we are going to have one more validation step. I think that's why we have developed this model. You can see clearly how this cells looks like in a 1 day after seeding, and then then we will wait for 3 days, which is the day 0 of untreated, and then you can see once you add some specific compound which targets a specific pathway, so people can easily find out how this ground state as well as this drug sensitive state are responding to the treatment. So I think using this model, like what we have did is actually we compared a similar same amount of cells in 2D as well as in the 3D, and using the same media components in both the cases, we just tried to see like if there is any difference in the activity when the cells are grown in 2D or when the cells are grown in a 3D model. So what we did, like we took a FDA approved and kinase inhibitor molecules as a screening. So we screened around close to 800 plus compounds in this. So I think this particular cluster, which is showing like the column 1 is like is the 2D model, and column 2 is a 3D model screen for the same compounds, because on the y-axis we have the name of the compounds, but I think all these compounds were clustered to the specific pathways which are in the x-axis. That's what we are seeing. But you can see there are some specific node. You can see some specific node, for example, like there is a differential activity which we are seeing here. So that is what we were looking for actually, because we wanted to select some compounds which specifically act more active in 3D, but in the same time it is not showing that similar activity in a 2D model. So I think our screen is also more robust. So we are seeing this here because it's showing a very clear correlation, was like really good. So then like based on that primary screen data, then we have further validated, and we found that so the PI3K pathway compounds and also the another related pathway, that's PI3K mTOR pathway, we have seen these compounds showed more response in a 3D model compared to the 2D model actually, because you can see this region which are we can see these two regions which is showing a clear effect on we can see the difference between 2D and 3D model. So what we did, like then we wanted to check like what kind of a gene expression profile which is changing when the same cell line, when it is growing in a 2D or versus the 3D model. So what we did, we took same cell line, like we took like two different cell line, which is like HN120. These are all primary patient-derived cell line. We took like HN120 and another one like HN137. So when we are looking for a different markers like SOX1, NANOG, vimentin, and other range of epithelial EMT markers. So we have seen like when cells are grown in a 3D model, this particular phenotype is matching with the which is the patient tumor, how it is behaving in the human system. So then what else we have also done, like some of the high content imaging. You can see the how this particular cell line has been stained for specific markers like CD44 in this case. So we have done this using our Phoenix, and you can see this. You can study basically how this particular marker is effective in this. So overall, what we have done, we have extrapolated this kind of a screening into more than 10 different cell line models. So I think this particular, the difference in inhibition scores between the 3D and 2D is giving us a clue that it will be better if we have a 2D model, and also we should have a 3D model in place to find out, because in a 2D model from this particular data, we can see these regions are specific for 2D. And These regions are specific for DUDI because these are like a PLK or a microtubule associated compounds, which is basically like a proliferation inhibitor. But like some of the specific pathway like mTOR, PI3K, and another major pathways, you can see this is like a number of compounds which are responding to this. So these are specific to 3D. So then like what we did, like we also did an in vivo validation of the highly active compound, and we found that whatever the hits we found in 3D model, which showed a more activity, it showed a clear tumor regression compared to the vehicle. So I think this is a very recent data which we have obtained. So I think based on this, actually now we have a model where we can understand, or we can study the number of drugs, and then we can give a output to say that actually, okay, this particular patient tumor cells is going to respond to any specific drug of what we are testing, right? So I think like based on this input from the different labs and other different places, so now there is an evolution of the cancer treatment. It's basically in a old paradigm. The doctors used to give a drug and say that okay, if it is not working, they will switch. So basically, it's like a trial and error prescribing sometime, because it doesn't suit every patient all the time with the right drug in the first choice. So the idea is to bring like efficient medical care, which is like a new paradigm which now every hospital is moving into. So what I think the advantage of this new paradigm is this is going to help in selection of the optimal therapy, and also this will also reduce the trial and error prescribing, and this will help to avoid adverse drug reactions, and this will improve the quality of life and also reveal the additional or alternative uses for medicines or the drug candidates. So I think what we do in our high throughput phenomics, as well as in a Ram Dasgupta lab here in GIS, is I think Ram is the one who is completely coordinating these efforts of precision oncology. So he had a lot of collaborations with the local hospitals here, which is National Cancer Center or SGH. So he has a close relation in working with Dr. Gopal Iyer and Dr. Leon Daniel from NCCS as well as GIS. So when the patient comes to the clinic, the patient tumor is implanted in mice as well as, and they were trying to expand it as a primary cell cultures. So this is like sometime we will directly derive this is PDPC, it is patient derived primary cells, or sometime then like we will grow them and we will also barcode them, and we are maintaining a biobank of this. And when these cells are multiplied in a larger scale, this will subject to high throughput screen. So then whatever the data we are seeing in this, which will be further validated in the PDX model, and so whatever the data output we are getting in this, the response data will be shared with the doctors to help or guide the treatment strategy for this particular patient. If he or like she, if they are coming back to the clinic with a relapse setting or with the kind of a resistant model, actually, I think that time this model will be useful. So the next slide. So here I am going to discuss like how our models are closely represent the patient tumor. So you can see our gene expression data. Like this is the patient direct the tumors which is which are exercised from the human. So we have subjected to gene expression analysis, and this is you can see which is 0.97, which is for the when the same tumor when it is growing in the mouse model, and this is our cell line, which is 0.95. So the advantage here is actually what we are seeing, the expression correlation between our models, three different models actually is completely highly correlated. And also we have tested this RNA expression. It's also similar across different models, which is close to 0.9 to 0.95 across different settings. So which is kind of giving us a confidence that I think the tumor complexity which we are seeing in the in vivo condition, which is still mimicking at the level of PDX, as also the patient-derived primary cell line models. So which is very, very useful for testing the multiple drugs on the particular cell line models. So we expanded the cells because in a lab. So then what we did, like we used to do, is like we used to test them against the set of FDA-approved anti-cancer drugs, and then our initial data showed some of the specific compounds which are working on a specific patient tumor lines here. So these are all like a different patients, like HN137, 120, 148, 159, and 160. So then we will, like what we did, like whatever the expressions we have seen, so that is also correlated with the molecular gene signatures which we found, like say some of the drugs which we found as a CDK inhibitor as a top hit, and we have also seen there is a dysregulation of this particular pathway which we can found in the same similar cell type. And also our subsequent validations in the same patient-derived xenograft model has shown that these compounds, whatever we found in the in vitro screen, is showing a very good correlation in the in vivo setting also. So this gives us a confidence that whatever the output which we are seeing from this data should be useful to the clinic, or like this can be a basis for a step for a translational research. So I think Dr. Ram and Gopal, when they were working on one particular patient, so they found that gefitinib can be more effective to this patient based on the data which we have seen in the lab. You can see this data, which is more effective for this particular patient-derived tumor line, and then we tested them on the in vitro model as well. So there also we have seen there is a tumor regression. So then based on the co-clinical settings at NCCS in Singapore, Dr. Gopal has tried this particular medicine, which we have found that which is showing a better response compared to the other standard of care, and we have also seen basically this whatever the data which we have seen in the in vitro condition is clearly worked in the clinic, at least in the N=1 co-clinical trial settings. So I think this is one of the best example of how we can develop this phenotype-driven precision oncology. So right now what we are trying to do is we are trying to increase the numbers. We are trying to increase the number of patients here, and then we are trying to add like a genomic and epigenome and gene expression and drug response data. We wanted to use this as a machine learned tool to find out, suppose if we do this for 25 different types of patients, maybe we will come back with a novel biomarker or like a novel signature that can predict again the drug response in a real time to the patients. So that is a hope for us to put more effort on phenotype-driven precision oncology at GIS. And so after that particular portion, so what I wanted to show is, so apart from our focus on 2D models and 2D models and then 3D models, and then how we use those models to do the oncology approach, where we can find out how the specific drugs are working for it. So here I'm going to show you some quickly, like what all the different other works which we are also doing, like using high throughput or a high content screening methods. I think what we are trying to do is, I think Dr. Judith Ko, she's our Computational Phenomics Platform leader. I think she put this she developed a database. It's like a Chemico-Pathway Profiler. So what it can does is, like, we are trying to create a bio-CV for the set of molecules. I think here we have worked close to around 15,000 compounds. We have tested them on like different pathways like ERK, Wnt, CMYC, and JNK, and all the pathways. I think you can see these clusters which are showing like the compounds against the specific pathway activity, so which will be useful for the smart selection of small molecules, or you can identify like how one similar scaffold is showing multiple effect. So this is an example of how this web interface can be helpful to the different scientific community. When they reach us, I think we can show them that, okay, like they can select some of the pathway which are upstream or downstream. They can select a very focused library based on this particular effort which we have put it on different compounds. They can identify the compound connectivity map, like say they can infer the putative target of uncharacterized compounds. So this will be an additional step which will help them to select the particular compounds. And this is another image-based drug screening which we have done, so in which the cells are labeled with different colors, and like we have tested them with a set of FDA-approved anti-cancer library. You can see like how these particular cell lines are reacting to the, because like we have also worked with, I think the previous study which I have discussed, it is with the oral squamous cell carcinoma, and this one is the colorectal cancer. So I think we are coming to the another one. This is the platform where I am leading here in Singapore, which is CHIP-GIS. It's called as Center for High Throughput Phenomics. I think our effort here is, I think we are working and supporting the different investigator-initiated programs in giving the expertise in assay automation and miniaturization. And also we will do a lot of chemical and functional genomics, and we do gain of functions and loss of function studies using CRISPR or si or shRNA, and we have different annotator libraries for small molecules as well as functional genomics. I think people who are interested to screen or like to know more about high throughput and high content screen, you can follow our website, which is chipgis.astar.edu. So this will give you the clue of what we are doing here on a day-to-day basis. And this is the companion computational platform, which is led by Dr. Judith. I told you because she's developing a lot of infrastructure development, because for all the high throughput screen, if you don't have an end-to-end solutions, the data will look really redundant, because I think all our high throughput screens data has been gone through this pipeline, and she will bring in this the ease of analysis and also the interpretations to move forward. I think this platform is very, very useful to give end-to-end solutions for all high throughput as well as all image-based screens. So with this, I am ending this presentation. I would like to acknowledge Dr. Ram and his team in GIS Singapore, and Dr. Gopal Iyer and his team in NCCS Singapore, Dr. Ian and Dr. Daniel at GIS and NCCS. And I would like to thank my team from Center for High Throughput Phenomics, and I would like to thank Dr. Huck. He's Executive Director, GIS Singapore, and I would like to thank Dr. Janardhan and also the PerkinElmer team for this wonderful support. So with this, I would like to end this presentation. I can take up questions now. Thank you.
Thank you, Dr. Periyasamy, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Periyasamy will answer as many questions as time permits. So our first question is, how do you determine which signaling networks or pathways are to be investigated or screened in your current study? For example, oncogenic, metastatic, anti-apoptotic, and immune modulation, etc.
Yeah. Hey, this is a very important question. When we do a phenotypic screens, sometimes we may not know the direct target engagement. I think the way we do this part of pathway analysis by high content biology approach using the Phoenix system that we have from the PE. So what we can do is, using this approach, we can derive the target protein engagement, and also we can study the changes in the cellular phenomena like nuclear morphology and membrane changes. We can track this information up to the per cell level data using this the deep imaging of high content imaging. So which is not feasible in say, for example, in the Western blot. Say you will take the whole lysate, so you won't be able to see what is happening in the per cell level data. So with respect to this phenotypic screening approach, I think we can do this by deep imaging called like high content imaging.
Okay, your next one is, will you be looking at any genotypic correlation in the patient based on the pathway-specific inhibitor identified?
Yes, absolutely. Yeah, because the whole idea behind this approach is to build a novel correlations between the individual drug response, which is corresponding to the gene expressions and the mutation status. I think when we are doing this as a pipeline, so what we are trying to do is we are also in parallel, we are doing the gene expression and the mutation status studies for that particular patients. So when we do this for more people, I think that will enable us to determine the novel biomarkers while we scale up the studies. Actually, that's an advantage.
Well, I would like to once again thank Dr. Periyasamy for his presentation. Do you have any final comments for us?
Yeah, I think this is one of the approach which a lot of people are getting into this right now. So I think people are using this particular the patient-derived tumor systems as a infer to move forward in the more advancing the drug development.
Thank you again. I'd also like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, PerkinElmer, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through October 20th, 2017. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye-bye.